Active and passive.
Phew, that hurt. Passive and active, what's the difference between them?
Active uses ATP.
Yep, active uses ATP and passive does not. What drives all passive processes? A concentration gradient, which is an unequal number of ions, molecules on either side of a plasma membrane. The first type is called simple diffusion. What happens during simple diffusion?
Molecules go straight through the phospholipid.
Goes straight through the phospholipid membrane because they have what kind of bonds holding them together? Non-polar covalent bonds. So our examples of things which go through simple diffusion are oxygen,
Carbon dioxide.
lipids, such as steroid hormones. Okay, now for things that either have a charge or are too large to fit through the membrane this way, we have another passive process called facilitated diffusion. So we are still using that concentration gradient, but these ions or molecules have to go through a protein channel to make it across. Our examples of things that move through the plasma membrane using facilitated transport are?
Things with charges.
Okay, so
Glucose, sodium.
glucose. Yeah, different color here. We got glucose. We've got sodium. We've got chloride, potassium. Let's see. Amino acids. Those are going to go through facilitated diffusion. And then we have a special type of diffusion. When it's water, we call it
Osmosis.
osmosis. And osmosis brought us to the topic of tonicity. So tonicity is basically talking about movement of water into or out of a cell, and it can change the shape of the cell. So we had three different types of solutions that we discussed. What were they? Hypertonic, isotonic, and hypotonic. Hypononic, tonic. Okay, so the first place that we need to start here is the baseline. What should your tonicity be inside of your body? What's your set point? What's the number?
285 to
Oh my gosh.
279 to 290.
Yeah, about 275 to 295.
I didn't know you were talking osmolarity. That's what the same.
Yeah, Jude, I don't know what you
That's what the saline solution is.
So what Jude said was 0.9, and we worked with this in lab. What was the 0.9?
Saline.
We called it normal saline, right? Why?
It wouldn't kill your blood cells.
It's the closest thing that we have to an isotonic solution in the medical facility. So hence normal saline, even though it is slightly hypertonic. Okay, so what would happen to the shape of a cell if we put it into a hypertonic solution? Crenation. What's that?
What's the unit for those?
Milliosmolar. And do not ask me to explain that.
No, that's fine.
I don't freaking know, and I don't care. It's just, that's how many particles dissolved in a certain amount of water. That's all we need to know from our perspective. What would happen to a cell put into the hypotonic solution?
Lice.
It's going to swell up and lyse. All right. Then that takes us to the next subject of primary active transport. What's our classic example of primary active transport in class? That is your sodium potassium pump, which pumps what?
3 sodiums out, 2
Two potassiums back in. Now, we can use the concentration gradient from primary active transport to do what?
Secondary.
To do secondary active transport. How does this work?
It brings in the left. Using leftover ATP to move stuff across.
Not using leftover ATP, because the only time we use the ATP is here. So we're creating this concentration gradient with primary active transport.
You basically just bring something else with you.
There you go. So what do we have to embed into the membrane? Over here we've got our sodium potassium
What's the
pump.
code?
And that's what creates this unequal distribution of ions, with more sodium outside, more potassium inside. And then what other molecule, what other protein do we have to put into this plasma membrane so that we can do secondary active transport?
Is that the co-transporter?
There you go. That's when we put in the co-transporter. And you can think about co-transport that we're going to transport two things at a time. One of those things is always going to be sodium, because that's where the energy comes from to bring whatever else into the cell. Because usually we're bringing something into the cell against its concentration gradient. And we can do things like glucose, we can do amino acids, we could do vitamins, whatever we needed. Where do we use secondary active transport?
Kidneys.
In the kidneys for reabsorption of the filtrate. And then our third type of active transport is called vesicular, because this involves vesicles, okay? Vesicular transport. And those are those big phospholipid circles, and we just transport large molecules throughout the body using this. If we're bringing a molecule into the cell, that process of bringing that large molecule into the cell with the help of a vesicle is called endocytosis. And if I am producing something inside of the cell that's a large molecule, and I want to dump it outside the cell, I call that process exocytosis. What is a very specific type of endocytosis done by some white blood cells?
Phagocytosis.
Phagocytosis. And they'll gobble up things like bacterium and get rid of them. All right, I think we might have just knocked that one out of the ballpark.
There's a question that was like, how is glucose transported? And I knew it was both of them, and I was like,
There's your concentration gradient, there's your simple, there's your facilitated, there's osmosis, there's your osmolarity, there's your tonicity stuff, primary, secondary, phagocytosis, exocytosis. And this is what we're going to do for our lab, I think next week or the week after. So there's that chapter.
Hey, we're gonna speed run
Okay.
all the chapters.
Well, it's so much easier when we're